Good morning, friends. I'm Dr. Vijay. Greetings from Vijay's classroom of biology to the viewers. So, in this class, we are going to deal with primer designing for DNA sequencing applications. So, there are many different kinds of primers. Some primers help in cloning purpose, and some primers help in DNA sequencing applications. So, in this class, we are going to deal in detail about the steps involved in designing the primers for sequencing applications, as well as I will show one demo using the software Serial Cloner 6.0 about how to design the primer with an example. So these are all the steps. So retrieve gene sequences of the target gene that runs from 5' to 3' direction from the NCBI database. You have to paste it in a Word file. So ideal primers should have a size of 18 nucleotide in length. So more than 18 nucleotide sequence of 5' target sequence is the forward primer used to amplify the target gene using PCR. 5' target 5' end of the target gene we copy around 18 nucleotide, more than 18 nucleotide in length, because chance of having repetition of this complementary sequence to this nucleotide or this particular primer is very less, because DNA is made up of 4 nucleotides A, T, G, C. So if we have primer which are 16 in length, there is a probability of the same complementary sequence to be repeated in any genome for multiple times. If we choose a primer that is less than 18 nucleotide in length, its repetition is more in a gene, and hence there will be non-specific amplification of the target gene. So we have to select 18 nucleotide from the 5' end of the target gene. We as such copy and we consider this as a forward primer, because this is going to anneal with the complementary sequence at the 3' end of the complementary sequence, and new strand will be synthesized. And we have to go for other step, that is reverse complement of more than 18 nucleotide sequence that is copied from the 3' target sequence. That is, the 3' end sequence of the target gene is taken. So we have one template, that is the target gene, from 5' to 3' direction it is running. It is a single sequence we have. So we generally copy the first nucleotide to the 20th nucleotide or 22nd nucleotide, and we keep as such, and we order for the forward primer. And for the reverse primer, because we have so only one sequence running from 5' to 3' direction, we just copy the more than 18 nucleotide length. We copy a particular sequence at the 3' end of this target gene, and we go for reverse complement generation. So the reverse complement to the 3' end of the target sequence is going to act as a reverse primer, because it is going to anneal with the 3' end, and it is going to amplify in the reverse direction, and it is called reverse primer. It is going to amplify the target gene in a direction opposite to the forward primer. And we have to do in silico PCR analysis using the forward as well as reverse primer along with the target gene sequence in Prime 3 software, BioEdit software, or Serial Cloner 6.0 or any higher version. So both forward and reverse primer should be of more than 18 nucleotide in length. We generally call it as 18-mer primer. So a primer containing 18 nucleotide is said to be 18-mer primer. Generally it is preferred to have 18 or more than 18 nucleotide in both forward as well as reverse primer to enhance the specific binding of the primer to the target site on the target DNA. So this is because the chance of repetition of the 4 nucleotides A, T, G, C in 16 combinations if 4-mer primer is used. If we take a primer of 4 nucleotide in length, then 4 to the 4 into 4. So 4 into 4 is 16. So this 4-mer primer can have 16 in 16 combinations. We can have a 4-mer primer to occur in the DNA, whose complementary sequence to occur in the DNA. There are 16 possibilities. So there will be annealing of this 4-mer primer at 16 places in the target DNA, which may render non-specific amplification. So in order to avoid that, we have to go for a specific amplification. We have to go with a primer that should have a size of 18 nucleotide or above 18 nucleotide. So this is because the chance of repetition of 4 nucleotides in 16 combinations, if we use a 4-mer primer, is more tending to non-specific binding of the primers to the DNA. Hence, it is in such a way that 18-mer primer is used to avoid combinatorial repetition of target sequence along the template DNA, tending to specific amplification of single target gene once at a time using a pair of primers. So both forward and reverse primers should satisfy the criterion that the GC content should be less than 40% and TM value should be between 50 degrees C to 60 degrees C. So these are all the primer qualities I am explaining. Though we copy these sequences, we should also check for the quality of the primer. It should have GC content less than 40%, then only EC annealing as well as EC denaturation is possible, and TM value should be between 50 to 60 degrees Celsius for both primers. It should not be at a fair distance between these two primers. Say for example, a 40 degrees C TM value for forward primer and a 60 degrees C TM value for a reverse primer may not give an average annealing temperature, because these two primers are not at all pairing, not at all falling within the optimum TM value as well as annealing temperature. If there is mismatch in the annealing temperature or TM value, then simultaneous annealing of forward and reverse primer to the DNA is not possible. So that's why both should have GC content less than 40%, because GC are connected by triple bond to the template DNA. These primers need higher energy for annealing as well as denaturation. So GC content should be less than 40% for ease of annealing and ease of denaturation, and at the same time TM value should also be between falling within 50 degrees C to 60 degrees C would be ideal. So both primers should satisfy the criterion that the hairpin loop, if suppose it is formed, must have TM value less than 30 degrees C, which means less than 30 degrees C, there is a chance for internal hairpin loop formation within the primer. Because in PCR we generally prefer a temperature more than 40 or 50 degrees C. At such temperature there won't be hairpin loop formation and all the primers are linearized, and it facilitates its exact annealing to the template DNA. But at less than 30 degrees C there can be chance for formation of hairpin loop, and that won't affect the PCR reaction. So it should have hairpin loops, if any, that can be formed within the primer, then that temperature should be less than 30 degree. Both primers should satisfy the criterion that delta G value, that is Gibbs free energy value for homodimer formation as well as heterodimer formation. Homodimer formation where two primers anneal each other, heterodimer formation where two different primers anneal each other, like forward and reverse. In case of homodimer formation, annealing between forward primers as well as annealing between the reverse primers separately. So that delta G value should be less than minus 6 kilocalorie per mole if homodimerization or heterodimerization is supposed to occur in the primer what we design. So these qualities we use integrated DNA technology page or any other software tools to check the quality. We paste the primer sequence and we look for hairpin loop formation, homodimer formation and heterodimer formation. The report comes in with the value delta G, but the ideal quality is homodimer formation or heterodimer formation should be less than minus 6 kilocalorie per mole. This energy is actually less than this energy is not possible within a PCR tube. So there is no chance for formation of homodimer or heterodimer if supposed to occur. Both primers should have the TM value difference of less than 5 degrees C, then only they can make a partnership in annealing with the template DNA. So there should not be much difference in the annealing between forward as well as reverse. The difference between the melting temperature as well as annealing temperature of forward and reverse primer should be less than 5 degrees Celsius, which is ideal for in vitro annealing steps in PCR. And there are variations in PCR, cloning PCR, where we design primers and we do PCR, and the PCR amplicons are meant for cloning. Colony PCR, where we take the single bacterial colony directly and then place it into the PCR tube, and we go for sudden heating and cooling for the template DNA to be released, and then immediate PCR reaction will be followed. So touchdown PCR commonly used to identify the DNA sequence from protein sequence by having primers with limited mismatches due to alternative codon sequence, and starting the PCR at high annealing temperature followed by gradual decrease in annealing temperature to promote amplification of specific amplicon. Long range PCR, amplification of larger fragments of DNA which are usually longer than 1 kb using special DNA polymerases or combination of enzymes with longer extension steps during the thermal cycling. Methylation specific PCR, identification of specific regions by using two primer sets, one recognizing cytosine where methylation is generally suspected, and other for thymine recognition to differentiate between the methylation states by analyzing the DNA treated with bisulfite to convert into cytosine, not methyl cytosine but to erase it. Multiplex PCR, performing PCR with multiple sets of specific primers within one tube that generate multiple amplicons for analysis, such as for mutational analysis or any pathogen identification, where pathogenicity island consists of multiple genes and multiple primers are simultaneously used in a single tube and they were amplified, but these primers should have a similar annealing temperature as well as melting temperature for successful PCR amplification. And in case of nested PCR, the other method, it uses two sets of primers in two sequential PCR runs, which the second primer set amplifies a sequence internal to the amplicon produced by the first set of primer, a method that is used to eliminate non-specific product amplification, and that is from the first primer set. So let us have a quick overview with a demo. So here is a gene sequence I am opening. So this is the gene sequence for alcohol dehydrogenase 2 gene from Saccharomyces bioanus. So herein we have one app we have, that is called serial cloner 2.6.1. So if you open this software, it will have an untitled sequence page already waiting for the target sequence. So we have a target sequence that encodes alcohol dehydrogenase enzyme. So we copy this. We can either copy only the sequence, or we can copy along with the FASTA format. So I am just copying this sequence. It is 1044 nucleotide in length. So here is an option next to the adapter, that is PCR. If you click it, it opens like run PCR, and here is a primer sequence which is 22 nucleotide in length. This is primer 1, that is being copied as a 5' end of the target sequence. So I am doing now itself instead of copying there. So here I am copying around 22 nucleotide in the 5' end. We are just copying, and then we are pasting it into the primer 1, that is forward primer FP. I am giving name Saccharomyces bioanus 1 primer name. It is given as 5' end this side and 3' end that side. In 5' end it is written as oligo tail not identical to the target sequence, where I am pasting, and it is 22 nucleotide in length. I am little copying around 1, 2, 3, 4, around 12 nucleotide at the 3' end of the primer that is pasted. I am doing cut paste, and then I am pasting in the segment identical to the target sequence. The segment non-identical to the target sequence are the editable sequences, where we can insert the restriction site of any restriction enzyme. As a result, if you do PCR reaction, the PCR amplicons will have restriction sites which can be restricted for sticky ends, and it can facilitate cloning. But now we are doing primer designing for just sequencing. We need amplicons, so we are not going for cloning. So that's why I don't edit anything like any insertion of restriction site into the primer sequence. So just I copied first 22 nucleotide from the 5' end. First 10 nucleotide I have pasted in the non-identical target sequence area of the primer, and then in the identical to the target sequence area I am pasting the other last 12 nucleotide. Anyway, its direction is 5' to 3'. Similarly, we go to the other end of the sequence, target sequence. Yeah, it is there, down till down. Yeah, there are around 22 I am selecting on the 3' end of the target DNA, and now I am copying. I will open another page in online, that is reverse complement bioinformatics.org. Even I will open in Google. So here is Google. You can type simply reverse complement, it will open, and now you can do clear. Now you keep cursor, paste the 3' end 22 nucleotide that is flanking at the 3' end of the target DNA. I am doing submit. So it is giving the reverse complementary sequence to the 22 nucleotide, and this is going to act as this one we have to copy as such. It is running from 5' to 3' direction. This we have to copy, and we have to paste it in the non-identical to the target sequence area of the primer 2. So primer 2 name is reverse primer, that is RP Saccharomyces bioanus. I am giving the name 1. This is the primer set 1, the reverse primer set that is going to pair with the forward primer. So that number indicates it belongs to that particular primer set. So I am just copying 12 nucleotide at the 3' end of the primer sequence that I have pasted, that the reverse complement sequence that I have pasted. 5, 6, 7, 8, 9, 10. Yeah, I am selecting and I do cut and I do paste. So there are 22 nucleotide. See now, what is the TM value? TM value ranges between 38 to 64, 38/64 for the forward primer as well as for the reverse primer. TM initial as well as final is 38/64, which means both TM value are same for forward as well as reverse primer. So now it is identical. And now because I have pasted the sequence in the untitled sequence 1 bar, I have to select the target DNA here. I can paste multiple sequences by opening new file, and I can say paste multiple set of target genes, and I can also select this select target DNA area, my desired target DNA. I can select my desired target DNA sequence what I have pasted here. So now it's a list. Here I'm going to select untitled sequence 1. If I open another sequence set, if I paste another sequence set, it is going to show as untitled sequence number 2. I can select any target DNA for the same set of primer to amplify. So now it is untitled sequence number 1 I have selected. Now I am going to click on, this is also 22 nucleotides. See the details are given below. Primer 1, its name 22 nucleotide, initial and final TM is 38/64, and here it is also reverse primer is also 22, TM value initial final 38/64. So maximum TM value it's showing, final TM value. So now I am going to click evaluate. So it's going to evaluate and it is giving a PCR product length of 1044. And now if I click run PCR, it's going to generate the amplicon with its sequence. That sequence will always be identical to the target DNA what we take, target gene what we take. It's a kind of in silico PCR we are testing to find out whether these primers work for PCR amplification or whether these primers can still work for DNA sequencing or not. To verify that, we are clicking run PCR and it has developed another fragment. This is the unsaved PCR product. Now I can click at it. I can go for save as PCR amplicon. This is also product size is 1044 nucleotide in length. See I am selecting and showing the number of nucleotides in the final product that is generated. Yeah, it is 1044 nucleotide in length. So the final PCR amplicon, if we go for sequencing, whose product length will also be 1044. So here we are not giving any primer annealing conditions or temperature. Actually we order the primer, we go for gradient PCR where we set a range of annealing temperature to find out the actual experimental successful annealing, and then we get amplicons later. But in silico PCR we give primers and we look for the probable complementary sequences that are found in the target DNA and its successful annealing within the system just in silico, and we are just running the PCR, and the PCR product whose sequence will also be 1044 nucleotide in length. Just for an example it is showing, but if we check the quality in integrated DNA technologies online page, we have to paste the forward primer and reverse primer. We have to look for its hairpin loop formation, heterodimer, homodimer conditions and all. If they all fall within the conditions what I have listed here on primer optimization, whose GC content should be less than 40% and TM value 50 degree to 60 degree C, as well as its hairpin loop formation less than 30 degree C, and delta G value for homodimer and heterodimer formation is less than -6 kilocalorie, as well as the actual difference between TM value for both forward and reverse primer should be less than 5 degrees C. If all these conditions uh, are ideal for the designed primers from the reports of integrated DNA technology online page, then we can order the primers in the integrated DNA technology site itself, and we can get primers. Once we get primers, experimentally we do PCR amplification with a range of annealing temperature, just by altering the annealing time and other things. We try to get the precise amplicons from it by varying the temperatures within a range. And once we get the fine amplicons, we go for sequencing, and the sequence reports should suggest the presence of the target sequence what we expected. And then we go for blasting and identifying the target sequence what we actually get after the sequencing process. So experimentally it is a highly laborious process, but an in silico verification before ordering, and in silico designing of the primer and verification would be very easy for any researcher to do. So thank you friends.